to help CD8 cells recognize antigens and start producing an army of fighters, killer or cytotoxic T cells. Helper T cells and cytotoxic or killer T cells, they have to work together. The cytotoxic cells depend on the helpers to get them working. Now, before they're activated, we just call them CD4 and CD8. In order to get them to mature and start producing an army of fighters or an army of helpers, they need to encounter their antigen. We saw that before with the B cells. They needed to meet their antigen before they would start breeding a clone and producing plasma cells. Now, these two types of T cells, the way they encounter their antigen is slightly different. One of them receives its signal from cells that have been infected with viruses. The other one receives its signal from cells that have just destroyed a virus. So helper cells are going to be created when a CD4 cell is met by a defense cell that has been fighting an infection, and it won. It killed the infection. It killed one of the pathogens that's causing the infection. Now that cell that's going to bring it news, here, I just killed this. It could be a macrophage. Remember we saw macrophages after they fight an infection and kill a few of the pathogens. We'll take little pieces of the pathogen and bring them to the lymphoid tissue and start showing those pieces to lymphocytes. This is what does it. It's showing off its trophies of the pathogen it killed. If this cell has killed, say, a bacterium, it could be showing a little piece of that bacterium to CD4 cells saying, look at this. Is this the antigen that matches your receptor? Does it match yours? Does it match yours? Trying to find a T cell, a CD4 T cell, whose receptor matches that chemical, that trophy from its kill. Often it's a macrophage that does this. There's also certain other cells in the body of these humans, like the dendritic cells embedded in the lower part of your epidermis. Your skin is a surface barrier. If it gets penetrated, microorganisms are going to have to cross through the skin to get deeper into your tissues. And so the dendritic cells there, in the upper outer layer of your skin, are also watching for pathogens. If they capture one and kill it by phagocytosis, they'll bring it to the CD4 cells in the lymphoid tissue and start showing it off. Here's my kill. Does this match your receptor, this trophy in line? We call those antigen-presenting cells. Cells that are holding up trophies of their kill. How do they do that? How do they hold up little pieces of their kill? Well, you might remember that earlier, when I was telling you about the natural killer cells, I showed you how cells will put little pieces of the chemicals they make on their surface. You might remember that those little pieces were being held in a special ribbon structure, a special clamp, called a class 1 MHC. MHC stands for Major Histocompatibility Complex. It's something that determines your tissue type, what these little clamps on the cell surface are holding. Now, in any cell with a nucleus, whenever it makes some proteins, it could be a protein of itself, or it could that it's made itself. It could be a protein that it was forced to make by a virus. We'll run that later. It'll take a few pieces of the proteins it manufactures, put them into class 1 MHCs, and mount them on their surface. Every cell in your body that has a nucleus does this. It's a way of saying, hey, these are the chemicals I make. Check them out. I'm healthy. Now, macrophages and dendritic cells and other antigen-presenting cells, they do this. They put the chemicals that they manufacture on their surface to say, hey, I'm healthy. Look at what chemicals I'm making. But they also make another kind of clamp. We call it class 2 MHC. And class 2 MHCs, they've been repurposed. Instead of showing off chemicals that the cell made internally, they're used to hold and show off, display, bits of whichever pathogen that cell has killed. So if this is an antigen-presenting cell, a